This lecture will be about some of the patterns of variation of respiration in trees and then some miscellaneous topics as well. You might remember some of these types of trait correlations from when we discussed the leaf economic spectrum. Here's an example where Peter Reich et al. Have plotted net photosynthesis rate. Note that this is per unit leaf mass, so it's per gram, as well as respiration per gram leaf mass. So these are all leaf mass bases, but at any rate, note that they've plotted the correlation between net photosynthesis rate and dark respiration rate for leaves of trees in many different places. And what you see is that there's a general and relatively tight correlation between those two, in that leaves with higher photosynthesis rates tend to have higher respiration rates. And that is related to, likely, the amount of protein in those leaves. Leaves that have higher net photosynthesis rates have more rubisco and other photosynthetic enzymes, and therefore there's a higher maintenance cost associated with those. So that's why those two things are likely related. I'd like to express this relationship in the form of the light response curve, just to tie this into some other things we've talked about. So if you look at the light response curve of net photosynthesis, where this is ppfd, which is photosynthetic photon flux density, that's in micromoles per meter squared per second. And this might go from 0 to around 2000. And this is P net photosynthesis rate, also in confusingly the same units, micromoles per meter squared per second. And let's say this is 0, so this is the 0 line. Let's say we have a leaf that's grown in the sun, and that leaf will likely have a light response curve that looks something like this. And so this is a sun-formed leaf. And then what color do you want to use? Let's use green. No. That's not colorblind friendly. Let's use blue for a shade-formed leaf. So the shade-formed leaf would have a light response curve that looks something more like this. So let's think about then the parameters associated with these two leaves. First, let's look at the parameters equivalent to these figures above, the net photosynthesis rate and the dark respiration rate. So by net photosynthesis rate, they're likely talking about light saturated net photosynthesis rate. So obviously in the sun-formed leaf, it has a higher light saturated net photosynthesis rate and a more negative or a higher respiration rate. In contrast, then, the shade-formed leaf has a lower light saturated net photosynthesis rate and a less negative or lower dark respiration rate. Again, associated with differences in sort of the amount of photosynthetic machinery. That sun-formed leaf is thicker, has more rubisco and other photosynthetic enzymes, therefore its maintenance costs are higher, and vice versa for the shade-formed leaf. I have a number of figures from O'Grady et al. 2010 in this lecture because their paper on foliage respiration in eucalyptus is just really good and demonstrates a number of the concepts that I like to talk about. So this is some more figures from that paper. In this case, showing R15. So this is respiration rate normalized to 15 degrees C, so measured at 15 degrees C, relative to height above ground for both dominant and suppressed trees. So a couple of things to note. First, as you get higher above ground, the respiration rate is higher. Okay, so for both of those, both dominant and suppressed trees, the respiration rate goes up as you go above the ground. But it looks like that relationship is different for the dominant and suppressed trees, in that for any particular height above ground, the respiration rate is higher in the suppressed trees than in the dominant trees. When they express that same variable against relative height, so 1.0 being the top of the tree, the relationship is basically the same. What that's telling us is that, let's think about, a forest where we have a dominant eucalyptus tree and a suppressed eucalyptus tree. What that tells us is that both of those trees allocate nitrogen within their tree crowns. The dominant tree, because it's taller, is going to tend to allocate nitrogen higher above ground just because it's taller. So it's going to essentially shift the allocation of nitrogen and therefore higher rates of respiration and higher photosynthetic capacity higher above the ground than the suppressed tree, which basically is only this tall. This is similar to the data that I showed you for Pacific silver fir, where I talked about allocation of new foliage growth in that dense stand, where those suppressed trees, they can only allocate resources as tall as they are tall. So this is another manifestation of that difference. But when you then sort of normalize that by relative height, they fall on that same relationship. In other words, the highest respiration rate is at the top of the tree, whether you're suppressed or dominant. So this is a nice demonstration of some of those concepts, in this case for respiration rate. A longstanding question in physiological research is the rate of respiration in leaves the same in the dark as in the light? It's actually a very difficult question to answer, because, especially with gas exchange, because again, when we measure gas exchange, we're measuring the combined effects of several processes: gross photosynthesis, respiration, and actually photorespiration as well. So remember, when we talked about photosynthesis, we said net photosynthesis equals gross photosynthesis minus respiration. And that sort of assumes that we know conceptually what that respiration rate is when we're measuring net photosynthesis rate in the light. So another way to think about that is, if we look at a light response curve, of net photosynthesis versus light. So remember, that curve looks something like this. And we know that the measurement of gas exchange at 0 ppfd right here, that's our estimate of respiration rate. And it's actually often called dark respiration because our estimate is that in the dark. But then the question becomes, does that same rate of respiration continue? I want to do red highlight. Does that same rate of respiration continue across that entire light response curve? As light gets brighter, do we still see that same rate of respiration at all these light levels? The short answer, as we understand it now, is no. The evidence shows that as light levels increase above the dark, the respiration in mitochondria actually becomes suppressed. And that's likely because there is abundant ATP produced in the light reactions of photosynthesis. And therefore, there's plenty of ATP in the leaf, at least, to meet those maintenance needs in the light. So conceptually, it makes sense that that happens, but measuring it has been difficult. So how has then the rate of respiration in the light been measured? Let's go back to the light response curve. Here they're labeling it par. That really means the same as ppfd. ppfd is a more proper specification of that. In the graph on the left, they're calling it CO2 flux, but that is the same as net photosynthesis. Okay. So again, that curve looks like this. This is 0, this is 2000. So this figure on the left is just an extraction of a part of this light response curve. Okay, so I just want to be clear where that's coming from. So they're zooming in on this really low light portion of the curve. So what they're showing, let's think about this, is when you measure this curve like this, you get these dots. So in the dark, you get this estimate of dark respiration, which is equivalent to this number right here. And then as you raise the light, that curve continues up and you continue up like this. But what they're pointing out is when you really zoom in, and if you measured at really fine increments of light increase, there's actually a point where, you know, we always think about this part of the curve as being linear, but there's a point where the slope of that linear increase changes, which is really interesting. So this really took very fine estimates of that light response curve. And what they found is that the slope of that changes. So at really low light levels, the slope is like this. But at higher light levels, the slope is less. And so this right here is the dark respiration that we've measured. So that's the respiration rate, the activity of mitochondria in the dark. But the reality is, in the light, if you extrapolate this shallower slope back to 0, you get this number, RL, which is an estimate of the activity of the mitochondria in the light. And that number is actually lower. So that's pretty cool, and that shows that in the light, the mitochondria are less active. Pretty neat. So these folks used those estimates, really fine measurements of light response at low light levels, and they found that, and in all of these, the shaded is respiration in the dark, and the light is respiration in the light. And basically it's two different species of, these are little shrublets in the tundra, but I think this applies to woody plants in general. And what they're showing is that across a range of nitrogen concentrations in these two different species, the rate of respiration in the dark is higher than the rate of respiration in the light across all of those conditions. So really neat study showing that respiration in the light is indeed suppressed relative to in the dark. They show a table showing a lot of the factors that likely contribute to that suppressed respiration rate in the dark, and there are a lot of explanations, including refixation of respired CO2 in the chloroplast. So the chloroplast basically grabs some of that CO2 that's emitted by mitochondria. Some of that CO2 diffuses into the chloroplast and is just refixed. It's never emitted from the leaf. And then there's a number of other explanations associated with downregulation of some of the respiratory enzymes that likely happens in the dark as well. Take-home message, again, is that in the light, respiration rates are suppressed. So a number of other papers in the literature have shown similar things as well.